Alzheimer's disease. Mechanisms and insights for clinicians. Introduction to Alzheimer's disease. Alzheimer's disease, AD, is the most common form of dementia, accounting for 60 to 80% of cases globally. It is a progressive neurodegenerative disorder characterized by memory impairment, cognitive decline, and behavioral changes. The hallmark pathophysiological features include amyloid beta plaques, tau neurofibrillary tangles, and widespread neuroinflammation. Let's delve into its molecular mechanisms and clinical significance. The molecular basis of Alzheimer's disease. 1. Amyloid beta plaques. The accumulation of amyloid beta, A-beta, peptides in the extracellular space of the brain is one of the earliest events in AD pathology. Formation. A-beta peptides are derived from the sequential cleavage of amyloid precursor protein, AP, by beta secretase, base 1, and gamma secretase. An imbalance between A-beta production and clearance results in aggregation of insoluble A-beta 42, forming plaques. Toxic effects. A-beta oligomers are highly neurotoxic, disrupting synaptic function and leading to neuronal apoptosis. They impair calcium homeostasis, induce oxidative stress, and activate microglia, contributing to chronic neuroinflammation. 2. Tau neurofibrillary tangles, NFTs. Tau is a microtubule-associated protein that stabilizes the cytoskeleton of neurons. In AD, tau becomes hyperphosphorylated, loses its normal function, and aggregates into intracellular tangles. Pathology. Hyperphosphorylated tau dissociates from microtubules, destabilizing axonal transport. Tau tangles block nutrient and neurotransmitter transport, leading to neuronal death. Spread. Tau pathology progresses in a predictable pattern, beginning in the entorhinal cortex and spreading to the hippocampus and neocortex, correlating with cognitive decline. Key pathophysiological processes. 1. Synaptic dysfunction. A-beta oligomers interfere with synaptic plasticity by disrupting long-term potentiation, LTP, and enhancing long-term depression, limited. Glutamate excitotoxicity results from excessive activation of NMDA receptors. 2. Neuroinflammation. Chronic activation of microglia and astrocytes in response to A-beta plaques releases pro-inflammatory cytokines like Illinois-1 beta, IL-6, and TNF-alpha. This inflammatory milieu exacerbates neuronal damage and promotes tau hyperphosphorylation. 3. Mitochondrial dysfunction. A-beta disrupts mitochondrial dynamics, leading to reduced ATP production, excessive ROS generation, and apoptosis. Dysfunctional mitochondria further exacerbate oxidative damage to lipids, proteins, and DNA. 4. Blood-brain barrier, BBB, dysfunction. A-beta deposition impairs BBB integrity, facilitating the influx of peripheral immune cells and exacerbating neuroinflammation. Risk factors and genetics. 1. Age. Age is the greatest risk factor for AD. The prevalence doubles every 5 years after age 65. 2. Genetic risk factors. Apolipoprotein E, APOE. The APOE epsilon 4 allele is the strongest genetic risk factor for sporadic AD. APOE epsilon 4 reduces A-beta clearance, promotes aggregation, and enhances neuroinflammation. Familial AD. Mutations in genes like AP, PSEN1, and PSEN2 are linked to early-onset familial AD. These mutations increase A-beta 42 production. 3. Environmental and lifestyle factors. Cardiovascular risk factors, hypertension, diabetes, and hypercholesterolemia. Sedentary lifestyle and lack of cognitive stimulation. Chronic stress and sleep disturbances. Clinical features of Alzheimer's disease. 1. Cognitive symptoms. Early stage. Memory impairment, particularly episodic memory, e.g., forgetting recent events. Intermediate stage. Disorientation, language difficulties, and impaired executive function. Late stage. Severe memory loss, loss of recognition, agnosia, and inability to perform daily tasks. 2. Behavioral and psychological symptoms of dementia, BPSD. includes apathy, depression, aggression, agitation, and hallucinations. 3. Neurological symptoms. Seizures and myoclonus in advanced stages. Pathological stages of Alzheimer's disease. The progression of AD can be categorized based on the spread of pathology and clinical symptoms. Preclinical stage. A beta deposition begins years before symptoms appear. Detected by biomarkers like CSF beta 42 levels and PET imaging. Mild cognitive impairment, MCI. Early memory loss without significant functional impairment. High risk of progression to dementia. Alzheimer's dementia. Significant cognitive and functional decline, corresponding to widespread neurodegeneration. Diagnostic tools for Alzheimer's disease. 1. Neuroimaging. MRI. Assesses hippocampal atrophy. PET scans. Detect amyloid deposition, using tracers like phlorbetaper, and glucose metabolism. 2. Cerebrospinal fluid, CSF, biomarkers. Decreased A-beta 42 and increased total tau and phosphorylated tau levels are indicative of AD. 3. Neuropsychological testing. Assess memory, language, visual spatial skills, and executive function. Treatment approaches. Although there is no cure for Alzheimer's disease, treatments aim to manage symptoms and slow progression. 1. Pharmacological treatments. Cholinesterase inhibitors. Increase acetylcholine levels by inhibiting its breakdown. Examples: donepezil, rivastigmine, and galantamine. NMDA receptor antagonists. Memantine reduces glutamate excitotoxicity. Monoclonal antibodies. Aducanumab targets A-beta plaques, representing a potential disease-modifying therapy. 2. Non-pharmacological interventions. Cognitive training, physical activity, and social engagement to maintain function. Behavioral therapy for managing BPSD. 3. Emerging therapies. Anti-amyloid therapies. Aimed at reducing A-beta production or enhancing clearance. Anti-tau therapies. Targeting tau aggregation and spread. Gene editing. Investigational approaches to modify genetic risk factors. Conclusion. Toward a better understanding, Alzheimer's disease continues to challenge clinicians and researchers worldwide. Advances in biomarker detection, understanding of molecular mechanisms, and emerging therapies provide hope for improved diagnosis and treatment. A multidisciplinary approach is essential to provide comprehensive care for patients and support for caregivers.